IMBRC Production presents a project by
Luna,
Florence,
Maria,
and me, Sarah. We're studying the stress responses of Methylus edulis exposed to the emerging contaminant sunscreen. Sunscreen is listed as one of the most concerning emerging contaminants regarding coastal waters, as it is a widely used product that makes sunscreen a source of directly introduced chemicals that have not been studied enough. Our aim is to see if two different kinds of sunscreen, one with a marine environmentally friendly label and one without, have an effect in the oxygen uptake and clearance of the model species Methylus edulis. We set the tanks with 3 replicates each, half of them with mussels and the other half without. We apply 4 different treatments, one of them is just seawater, 2 others are diluted sunscreens and 1 with a hand treatment as a control. We first collected our samples in the Bay of Jarneux from a concrete pier near the marine station. After some time looking between the algae, we finally found the mussels, to be precise Methylus edulis. We started collecting them randomly, detaching them carefully with a diving knife from their substrate while snorkeling. And after having enough mussels to start our experiment and enjoying Jarneux's underwater life for a bit, we head out of the water. Here you can see us super happy with our buckets full of mussels. Thanks to Nico for joining us in our brief but nice fieldwork. We get the buckets with our mussels and head to the wet lab. Afterwards, we put them in a tank, letting the mussels acclimate with the constant temperature and flow. We proceed with cleaning the mussel shells, scraping the surface carefully with a knife, then weigh and measure them. Meanwhile, we prepare our 24 tanks of 2 liters each, connecting the silicone pipes for the bubbling. We fill a big tank with cold salt water for the treatment with the effect of our bare hands. But first, we have to wash them really well. Then we spend 20 minutes with 3 pairs of hands dipped in 6 liters of water. Afterwards, we start preparing the sunscreen treatment. We calculated the amount, which resulted in 1 gram per 2 hands. We applied in our clean hands and then we dipped them in water once again. Once we filled all the tanks, we have everything set to start. Here we are bringing the mussel to the climate room to keep a constant temperature during the experiment. We place the mussels into their new home for the next few days and we measure the water temperature and salinity. The experiment can finally start. We start the oxygen measurements, covering the tank openings with parafilm and tinfoil, preventing oxygen exchange. Then we measure the oxygen dissolved in the water with an oximeter. We also take water samples for later analysis with the spectrophotometer. During the night, we keep the mussel with constant bubbling and we repeat the oxygen measurements at least twice a day. Once we finish, we clean the tanks and weigh the mussels again. Now, after some very intense analysis, we have some results and we first focus on the short-term ones. While there were changes in oxygen concentration for the green sunscreen, it seems that with the conventional one, in the first 2 hours after exposure, the mussels stop the aerobic respiration and might change to an anaerobic metabolism, which might be due to stress reaction. We are also studying the long-term impact. Over a period of 3 days, we took measurements in the morning and evening to compare and assess changes over the course of each day. First of all, oxygen levels inside the tank decreased over the days. This means that all the mussels breathe, but at different rates depending on whether or not they are exposed to sunscreens. Ultimately, we couldn't show with a certainty in our experiment that different sunscreens have an effect on Methylus edulis. So there's room for improvement for our experimental design and future research. Last but not least, to celebrate the end of our research project, we released the mussels that had no sunscreen treatment back to the sea where they belong.